Good afternoon, everyone, and welcome back to our virtual clinical educational grand round. Today's presentation is from our Faculty of Pre-Med, which presents every Monday. I am your host, Ms. Shinondo, here as part of Corporate Communications. First of all, I would like to acknowledge the presence of our professorial team, Professor Njelesani, Professor Lambert, Professor Mudenda, Professor Mwawa, as well as Professor Mulenga, Professor Emaus, Professor Gilani, Professor Obeten, Professor Etta, Professor Viemba, Professor Kasonka, as well as Professor Kumar and Professor Kasolo. Today's topic is transport across cell membrane. Now, please be aware that these presentations happen every Monday from the Faculty of Pre-Med. All the other days of the week equally have their own faculty. Monday, Pre-Medical Sciences. Tuesday, Preclinical Sciences. Wednesday, Research Presentation. Thursday, Grand Round presentations. These are presentations from our students across various faculties, notably MBCHB, while they present their experiences and findings during their ward rounds. Then Fridays are special presentations awarded to our faculties of Pharmacy, Health Sciences, and Nursing. The reason behind, or rather the motive behind these presentations is skill set building within our students. Skills such as long case presentation, public speaking, as well as presentation making. Whether that is doing the actual research, as well as learning how to compile and translate their information. This is an excellent opportunity, not just for our presenters, but also you who are in attendance. This is also your chance at some point during question and answer for you to ask all the relevant questions or make any observations. Who knows? Your question or comment can open the doors to further discussion, and that is how we learn. Without any further ado, I would like to invite our moderator, Dr. Chirwa, to briefly introduce the topic and begin the presentation. Dr. Chirwa, the floor is yours.
Okay, thank you very much. So the living component of the human beings are the cells. They take up materials from the external environment. The materials move in a variety of different ways. Our different presenters are going to talk about the different ways in which materials move. Now, what happens when we have a defect? What happens if a cell cannot take up one or two components that is required for the normal cellular functions? Again, our presenters are going to cover one or two conditions such as diabetes. We are going to find out what exactly goes wrong in that situation. Transport across cell membrane is part of our curriculum. We covered it in the opening week of the semester. Because it is part of our curriculum, it is going to appear in the quiz, test, and final examination. To our audience, we ask you to be very interactive. Please ask as many questions as you can. Our presenters are very much ready for you. Where there may be need, I'll be coming in. So over to you, our presenters.
Thank you. Good afternoon, everyone. My name is Moses Mogala from the Faculty of Political Sciences, and I'm here with my colleagues. It's a combination of first and first-second students. So as earlier mentioned, we'll be discussing transport across the cell membrane. So the first will go first and discuss the structure of the cell. Then second will come in to discuss transport and the conditions associated with transport across the cell membrane. So we'll go into it with the first to do the first part, which is the structure of the cell.
Good afternoon, everyone. My name is Nene Piri, and I'm the first speaker of this presentation. So today we'll be presenting on transport across the cell membrane. So we'll start with the introduction. The survival of every cell depends on the maintenance of an intracellular content unit for that cell type, despite the remarkable different composition of extracellular fluid surrounding it. This difference in fluid composition inside and outside a cell is maintained by the plasma membrane. A plasma membrane is an extremely thinner layer, the lipids and proteins that forms the outer boundary of every cell and encloses the intracellular contents. So we have the structure of the cell membrane. The cell plasma membrane surrounds the cell. It is made up of 55% proteins, 42% lipids, and 3% carbohydrates. It allows some substances to pass through it and excludes others, which means it's selectively permeable. It is 6 to 10 nanometer thick. The outer and inner membrane surfaces are different. So as you can see, we have the structure whereby we have the glycoproteins, lipids, hydrophilic heads, hydrophilic tails, and so on. Structure of the cell membrane. The phospholipids make up 75% of lipids, while cholesterol and glycolipids constitute 20% and 5% respectively. Phospholipids are the main component of phospholipid bilayer and consist of hydrophobic tails face inward and the hydrophilic heads face the water. Cholesterol maintains proper membrane fluidity. Structure of the cell membrane phospholipid bilayer. As you can see, we have the extracellular space, the lumen of cell, the phospholipid, polygroup, phosphates, and so on. The diagram is here.
My name is Birik. We're going to continue from one my colleague. We're going to look at the structure of the cell membrane, protein which are found in cell membrane. So there are two types. The integral membrane protein is inserted into the membrane hydrophobic region that just adjacent to hydrocarbon pools. The other type is the peripheral membrane protein attached to either the inner or outer membrane surface. The functions of the proteins which are found in the cell membrane: transporting of materials across cell membrane, enzymes, receptors of chemical messengers, meaning they detect, and then identification of cell recognition. So cells can identify themselves through proteins. Attachment, membrane inside the skeleton and intracellular junctions. As we were looking at the proteins, right now we're looking at carbohydrates. We can tell from the protein that the carbohydrates are located on the outside surface of membrane and are key to cell recognition. So same as the proteins, the carbohydrates are also used for cell recognition. So glycolipids with sugars, glycoproteins, proteins with sugars, then transportation antigens. So varies greatly among individual species. Organ transplants require matching cell markers, and immune suppression. Fluid quality of plasma membrane. In a living cell, membrane has fluidity as salad or unsaturated hydrocarbon ions increase membrane fluidity. Phospholipids and proteins literally. Phospholipids move very rapidly, proteins drift in membrane slowly. Cholesterol alters fluidity of membrane, decreases fluidity at warmer temperature, which is 36 degrees Celsius. Increases fluidity at lower temperature, 87 degrees Celsius. So as you can see, the diagram, fluid quality of plasma membrane. We can see hydrophilic region of proteins, hydrophobic region of proteins, also the phospholipid bilayer. functions of the cell membrane. Separate cells from non-living things, form most organelles and partition cells into discrete compartments. So the cell membrane separates living things, separates the cell from non-living things of the environment, regulates passage of materials in and out of the cell and organelle. Membrane is selective permeable. No wonder it's called the selective permeable. It regulates passage of materials in and out of the cell and organelle. Receives information that permits cells to sense and respond to the environment. So it receives information that a hormone that permits the cell to sense and respond to the environment, hormones, growth factors, and neurotransmitters. The other part of the functioning of the cell membrane is for communication with other cells and organisms as a whole. The surface protein allows us to recognize each other, adhere, and exchange material. Transport across cell membrane. Non-polar hydrophobic molecules dissolve into membrane and cross with ease. The smaller the molecule, the easier it can cross. Example, oxygen, hydrocarbon, steroids. So the non-polar, from the definition of from saying, we can say the smaller the molecules, the easier it can pass. Polar, hydrophilic molecules, smaller polar and charged molecules can pass through easily. For example, water, carbon dioxide. Large polar and charged molecules pass with difficulty, meaning they can't pass due to the bigger in size. Ionic hydrophilic molecule, charged ions or particles cannot get through, such as sodium, potassium, and chlorine.
Good afternoon, everybody. My name is Anila Indlo. I'm going to talk about the passive processes. So the passive processes, these are the methods used to transport in. These are the methods which are used across cell membrane. They do not require the use of energy. They do not require the use of energy. These processes These in this process, substances move down their concentration gradient, meaning they travel from a concentration of high concentration to a region of low concentration. In these processes, we have three types of passive processes, which are simple diffusion, facilitated diffusion, and osmosis. Under simple diffusion, these are the simple diffusion membrane. Under simple diffusion, passive types of transfer describe the movement of substances down the concentration gradient and do not require the use of energy. Simple diffusion is the net movement of substances from an area of high concentration to an area of lower concentration. Simple diffusion means that the kinetic movement of molecules or ions occur through the membrane opening or through the intermolecular space without any interaction with the carrier protein in the membrane. Examples of substances that can be transported through lipid barrier includes oxygen, carbon dioxide, and nitrogen gas. Other solids that get transported through lipid barrier are fatty acids, steroids, and phytosoluble vitamins. The diagram which is shown there is a simple diffusion. In where there is direct movement of molecules in the lipid barrier without the help of the protein. It occurs in the membrane opening or intermolecular space, relying only on the energy, on the natural kinetic energy. There we can see the guts close and guts opens of the simple diffusion. Facilitated diffusion Facilitated diffusion is the diffusion of solute through channel proteins in plasma membrane. Facilitated diffusion is also a carrier-mediated diffusion because a substance transported in this manner diffuses through a membrane to other sites using a specific carrier protein. The molecule to be transported in enters the pore and becomes bound, and conformation or chemical charge occurs in the carrier protein, and the pore opens to the opposite side of the membrane. Among the most important substances that cross cell membrane by facilitated diffusion are glucose and most of the amino acids. Under this diagram, we see osmosis, the osmosis diagram, the bounding point transported molecules, carrier proteins, and conformation changes, and the release of binding. Osmosis is defined as the process of net movement of water caused by concentration difference across cell membrane. If pure water is put on one side of the cell, the membrane and the solution of sodium chloride on the other side. Water molecules pass through the cell membrane with ease, whereas sodium and chloride ion pass through only with difficulty. The diagram which is shown shows the water and sodium chloride solution and the movement of water molecules.
Good afternoon, everyone. My name is Amara Joseph, and I'll present on active transport, active process. So active processes utilize energy mainly in form of ATP to transport substance across the cell membrane. These include active transport, bulk flow, exocytosis, endocytosis, and transcytosis. Active transport across the cell membrane. Active transport is the movement of solids against a gradient and requires the expenditure of energy usually in the form of ATP, which is adenosine triphosphate. Transport proteins in the plasma membrane transport solutes such as small ions, potassium, chlorine, hydrogen, amino acids, and monosaccharides. These proteins involved with active transport are also known as ion pumps. The protein binds to a molecule of the substance to be transported at one side of the membrane. Then it uses the release of energy, ATP, to change its shape and leave it on the other side. The protein pumps are specific. There is a different pump for each molecule to be transported. Protein pumps are catalytics in the splitting of ATP, ADP plus phosphate, so they are called ATP pulse enzymes. So here is a diagram of active transport. So here we have the potassium there from the other side, 2K. It moves through the gradient and it uses the energy ATP pulse. Bulk flow. There are three main types of bulk flow: exocytosis, endocytosis, phagocytosis. Under endocytosis, we've got phagocytosis, phenophytosis, receptor mediator endocytosis, and the third one, which is transcytosis. Exocytosis, described as the bulk movement of substances out of the cell into extracellular environment by fusion of secretory vessels with the plasma membrane. The material for secretion is packaged with intracellular transport vessels, which move towards the plasma membrane. When the vessels and plasma membrane come into contact, the lipid molecules of the vessel and plasma membrane bilayers rearrange themselves so that the two membranes fuse. The fusion of these lipids bilayers require the cell to expend energy in the form of ATP. Following fusion, the vesicle contents are released into the outside of the cell. An example of this process is release of digestive enzymes by pancreatic acid into the pancreatic duct for transport to the small intestine. Another example is release of neurotransmitter into the synapse fluid by neurons. So below is a diagram showing the cytoplasm. When it is carrying the proteins, it more engulfs them, then it will attach to the layer, and then it will open up, and then it will release them. Tear glands export salty solutions. Pancreas uses exocytosis to secrete insulin. So the diagrams here are just a continuation of what I was saying in the previous one. The first one is the vesicle near the cell, the plasma. The cytoplasm is there, the circulatory protein, circulatory vessel, the vessel membrane, and the intracellular vessel.
Greetings to you all. My name is Tanera Jayun, and I'll carry you through endocytosis and transcytosis. In endocytosis, extracellular macromolecules and large particulates matter are packaged in a vesicle that forms at the cell surface of internalization into the cell. A small area of plasma membrane folds inward to form a pocket, which is also known as an invagination, which depends and pinches off as the lipid bilayer fuses. New intracellular vesicle is formed containing material that was formerly outside the cell. Three types of endocytosis include phagocytosis, pinocytosis, and receptor-mediated endocytosis. So here we have a diagram that shows us the uses of the endocytosis uses vesicles to move substances into cell. So in A, we have the flow of phagocytosis, and then in B, it's pinocytosis. In C, it's receptor-mediated endocytosis. Phagocytosis, which is also known as cell eating. It is a non-specific process that occurs when a cell engulfs a large particle external to the cell by forming membrane extensions, also known as pseudopodia, or false speech to surround the particle. Once the particle is engulfed, it is packaged within an enclosed membrane sac. The contents of the vesicle are broken down, or also known as digested, after it fuses with the lysosome, which contains specific digestive enzymes that split large molecules into smaller ones. Phagocytosis is the process by which bacteria, dead tissue, or other bits of microscopic material are engulfed by cells, such as polymorphonuclei, which are also known as multi-lobed nucleus, leukocytes of the blood, and macrophages in connective tissue. And here we have a diagram that shows us the flow of endocytosis. So in the first part up there, we can see that the pseudopods are extended so that they can engulf the microbe. And then once the microbe is engulfed, it is attached to the receptor there at the bottom, and then it moves to form phagosomes. And after the phagosomes are formed, the phagosome and the microbe fuse together, which forms lysosomes and phagosomes. And then the next process is digestion of lysosomes by enzymes, and then the residuals of the body, and then the waste remains as residual body, and as time goes on, the cell will excrete them. Pinocytosis, which is also known as cell drinking. This is a non-specific process that occurs when the cell internalizes a very small droplet of extracellular fluid into tiny internal vesicles.
Within the cell, the vesicle fuses with the lysosome where enzymes degrade the engulfed solids. The resulting smaller molecules, e.g., amino acids and fatty acids, leave the lysosome to be used elsewhere in the cell. Pinocytosis occurs in cells of capillary wall, where vesicles fill the fluid, droplet containing small solids from the blood, carry this droplet to the other side of the cell, and then expel its contents outside the capillary wall, e.g., transportation of fatty acids from plasma into adipose tissue, also known as fatty cells. And here we have another diagram of pinocytosis, where it starts with formation of vesicles, and then it goes to the fluid and dissolved solids of vesicles. And then the lysosomes attach with the vesicles to form digestion lysosome enzymes, and that's the end of the process. Receptor-mediated endocytosis. This is movement of specific molecules from the extracellular environment into a cell by way of newly formed vesicle. It begins when molecules in the extracellular fluid bind to their specific integral membrane, protein receptors. It is considered a specific mechanism because the endocytosis is stimulated by the binding of the specific molecules to their specific membrane receptors. The receptor proteins then cluster in one region of the membrane to begin the process of endocytosis. The plasma membrane housing the bound specific molecules from the extracellular fluid folds inward to form a pocket, which we said is an invagination. In this membrane, pocket depends and pinches off as the lipid bilayer screws. We have a diagram that shows blood cholesterol that is taken up by liver cells through receptor-mediated endocytosis. So in the diagram, we can see that we have protein, cholesterol, and the cholesterol is taken into the phospholipid outer layer, going into the cytoplasm. An example of receptor-mediated endocytosis occurs when human cells contain receptors that bind to internalize cholesterol, which is required for new membrane synthesis. Cholesterol travels in our blood bound to proteins called low-density lipoproteins, in short LDLs. LDL particles bind to LDL receptors in the membrane. And we have another diagram that shows the flow of the cholesterol in our body. So in the first, we have the receptors of LDL samples, and we go to vesicle formation, and then from there we go to, I'm so sorry, I cannot see that far.
We have transporting fusion with endosome, recycling of the receptor degradation. Okay.
And then number 4, we have fusion with endosomes. They are transported to the vesicle. And then we go to number 5, recycling of receptors to plasma membranes. And then we go to number 6, which is degradation in lysosomes. And we have endosomes, transport vesicles, digestive enzymes, and lysosomes. Transcytosis. This is transport in vesicles, which involves successive movement of a substance into, across, and out of a cell. In this active process, vesicles undergo endocytosis on one side of a cell, move across the cell, and then undergo exocytosis in the opposite side. Transcytosis occurs most often across the endothelial cells that line blood vessels and is a means for materials to move between blood plasma, interstitial fluid, e.g., movement of maternal antibodies across the placenta into the fetal circulation. Here we have a diagram of transcytosis. It starts we have the bilayers and then diffusion through the lipid bilayer. And then from there, we show the passive transport. We have the facilitated diffusion, which goes through the lipid bilayers, and we also have active transport, and it is energized by ATP.
Good afternoon once again. My name is Moses Mugala, and I represent on the common disorders associated with transport across the cell membrane, as well as the conclusion of this presentation. So among the conditions, the disorders, we have cystic fibrosis. So in cystic fibrosis, mainly what's happening is that the cystic fibrosis transmembrane regulator, which is a chloride channel protein, is defective. So because it's defective, it cannot properly control the flow of chloride out of the cell. So when that happens, the end result is that there's also poor inhibition of the epithelial sodium channels, leading to a lot of sodium entering the cell. Now, we know from science that where sodium goes, water goes as well. And because there's a lot of sodium entering the cell, water leaves the lumen as well and enters the cells. Okay, so next one we have diabetes. This one comes in two types of diabetes mellitus type 1 and diabetes mellitus type 2. So a brief background of what happens under normal circumstances in the transportation of glucose across the cell is that when glucose enters the bloodstream, is acid that will activate the glucose transporters to facilitate for movement of glucose to enter the cell and leave the bloodstream. But in diabetes, insulin is not produced. In type 1, insulin is not produced because the cells responsible for producing it are damaged. So because that does not happen, the glucose transporter is not activated, leaving the levels of glucose high in the blood and leading to diabetes type 1. Then in type 2, there's a poor signaling or poor communication between insulin and the glucose transporter. So in type 2, insulin is produced upon glucose entering the bloodstream, but because of that poor communication, you find that not much of the glucose enters the cells, and subsequently it still leads to large amounts of glucose being in the bloodstream and leading to diabetes type 2. Okay, so the third one is Leber syndrome, which is basically a kidney iron disorder causing hypertension because of excessive sodium reabsorption. So again, in Leber syndrome, there's poor regulation of the epithelial sodium channels, which leads to a lot of sodium entering the cell. So when a lot of sodium enters the cell, to try to balance that charge, what happens is that a lot of potassium will exit the cell, and also hydrogen ions will exit the cell. So when that happens, it leads to three main things. First, we will have large amounts of sodium into the cell. We'll have low potassium in the cell, and we also have low hydrogen ions. The end result is hypertension, hypokalemia, and metabolic alkalosis. The fourth one is renal tubular acidosis, which also comes in two types. There's distal and there's a proxim. So here there's impaired hydrogen ion transport in the kidneys, resulting in the blood becoming too acidic. So here what happens is there's a lot of hydrogen ions entering the cells and leading to the acidic environment. Then you have Bartter syndrome, which affects co-transporter in the kidneys, impairing reabsorption of sodium, potassium, and chloride. So the end result, because of the co-transporter being defective and poorly regulated, it leads to electrolyte imbalances. The sixth condition is a long QT syndrome, which will be seen on an ECG and electrocardiogram. Normally, the action potential in the cardiac muscle is influenced by the exchange of ions across the cardiac muscles. So in a long QT syndrome, what's happening is that there's poor timing or improper entry and exiting of these ions, which leads to this condition we're calling the long QT syndrome. In conclusion, understanding transport across the cell membrane is key to understanding cellular function, homeostasis, and overall organismal health. because the process governs the movement of essential molecules like nutrients, gases, and waste products, enabling cells to survive and function properly. To understanding various diseases and disorders, including cystic fibrosis, diabetes, and neurodegenerative diseases like Parkinsonism and Alzheimer's. And those are our references. Thank you so much.
Okay, thank you very much to our presenters. Ms. Shilondo, over to you. Hello, Ms. Shilondo. Okay, to the people who are present, we are trying to do a roll call. Kindly, you can unmute and tell us where you are listening in from, or you can type it in the chat box. If you are Chalala campus, you say, oh, Chalala campus, New Chalala campus, Lusaka, or anywhere where you are listening from. Okay, we have someone from Chalala campus. Thank you very much. The others, where are you following from? Miss Shilondo, are you still with us?
Yes, I am. Apologies. I had some technical difficulties. Dean, apologies. Could you please repeat the last thing you said? I was
asking,
disconnected.
we are taking the roll call. Try and find out where our listeners are following us from.
Okay, wonderful, wonderful, excellent. Thank you. So we have Nawa listening from Chalala campus. Welcome. Do we have anybody else? You can either raise your hand when I mention where, or you can write down in the comments where you are tuned in from. We have Gumi, that's Open Distance Learning, Chalala campus. You are listening from Kafue. You are more than welcome. Excellent. Do we have any others listening from our Chalala campuses? Any of them? What about Mutandwa? Do we have any from Olympia? What about Chipata? Do we have any from our Chipata sites? Please do not hesitate. Let us know. Because I see we have a number of people in attendance. Please let us know. This is your time. We have McCarlin. This is Jane Forster, Open Distance Learning, Chalala campus. Welcome. We have Chisanga Lengwe, Open Distance Learning again, Chalala. This is good. This is good. We have a lot of Open Distance Learning this time. Well done. You are representing your demographic well. In the meantime, while we have other names coming through, the floor is open. Question and answer session. Please do not hesitate. So this is your chance to get a better understanding of what our presenters have just unveiled to us. Also, job well done, presenters. We have Lambongo listening from Mpika. Welcome, Open Distance Learning again. Excellent, excellent. You can either ask your question by raising your hand, the floor will be given to you, or you can write your comments, questions, and your observations in the chat, and they will be read out for you. Once again, this is an open platform. This is a chance for you to ask whatever questions or share whatever observations, and definitely this will open more doors to further discussion as well as more observations. These may be presentations, but this is still class in session. And since I'm looking at how many we have in the house, the longer you keep quiet, I will start calling names. So please do not hesitate by all means. And for those of you who just need a bit of a reminder why we have these presentations, these are skill set building within our students. As an institution, as Lusaka Apex Medical University, we ensure that our students do not just walk out educated, but they walk out exposed. This is also why we have an international panel of professors on our team. We have not only some of the best, which are based here in Zambia, but we have others from Norway, Pakistan, and other places. So once again, the more doors you open through what you may say, the more feedback and the deeper the depth of knowledge and or explanations you shall receive, not just from the presenters, but also from our team of professors. We ensure that here you get the exposure. Madam Muntanga, do you have anything to share? A simple yes, no, this feedback. This is a chance.
No.
All right, wonderful. What about Vuniwe? Anything? Madam Vuniwe? What about Nene? Well, it seems we have a shy crowd today. I would like to invite any of our members from the professorial team. Do we have any present with us in the boardroom?
Yes.
Wonderful.
Yes, we do. We have top members.
Pardon?
Thank you. Madam Chinondo, can you hear me?
Yes, welcome, Professor. Though unfortunately, I cannot see you.
Oh, we can see you clear. Thank you very much. I first of all, I want to thank Dr. Chilwa for a well-organized grant round. And for pre-med students, this is well done presentation. But what I want you to do, maintain your notes as you rise on the ladder of medical education. Whatever field you choose, those who become doctors, pharmacists, nurses, you name it. But keep your notes, because you need this knowledge. You need this knowledge, and I'm happy you're already applying the knowledge. So in clinical practice, for example, the brain has to pick up oxygen, fresh oxygen, for it to work well. And in your medical practice, you will know that the brain cells that are demanding oxygen so much. You talk about oxygen, and it's 4.5 mils of oxygen every minute for every 100 grams of brain tissue that the brain is really wanting to get. But it also throws out the carbon dioxide. So we have transport in oxygen, glucose. Glucose 4 grams, 4 grams, 4 milligrams every 100 milligrams of brain and for every minute. Again, it's another statement you need to know. Glucose. Because the less glucose it is, patient will fail. Hypoglycemia for all. So you already applying your knowledge on diabetes, glucose uptake, but metabolism will release byproducts like what? Carbon dioxide. Where should it be carried? Circulatory system will take it to the lungs. Deoxygenated blood will carry carbon dioxide from a brain, for example, to the alveoli in the lung. I'm reaching there with deoxygenated blood. It has to release the carbon dioxide and it pick up fresh oxygen. But some of the cells get damaged, red cells. So at the alveoli there, damaged cells are picked. You mentioned the word phagocytosis. Phagocytes are ready there to pick damaged cells and also the particles that we breathe in from atmospheric air. Those have to be removed. Now, you look in your family, you find elderly people, when they wake up, they cough out, especially smokers. They cough, and you watch the sputum. That sputum is full of either phagocytosed damaged cells and particles that need to be removed out. So by coughing, you can tell what kind of sputum it is. In those with, for example, congestive cardiac failure, they will cough more red cells, damaged red cells out. So the color of the sputum helps you to understand what is in that sputum. So you are dealing with patients even at your early stage. You are understanding. Cell membrane is very key. It allows in, it allows out, sometimes very selectively. For example, as you say, the smaller molecules are easier to cross. It is made like that. The larger molecules are prevented. The charged molecules, they are, again, cell membrane stops there so that they can cause problem in the intracellular environment. So the cell membrane structure, the way I presented it, I'm very happy. It's really sustaining health of the cell. Cell health is determined. Because if you just bring everything, you can damage the cell. But also use it for selective treatment. Things like a patient who has got problems that require certain antibiotics to cross over and treat intracellular environment. So you find certain antibiotics, you need to know them, which ones are going to be suitable for treatment of such a condition. You consider the cell membrane. What about the liver? You mentioned the liver. The liver is very useful because the bilirubin, that is the output of damage of, bilirubin, a red cell. It has to be picked by liver cells, conjugate bilirubin, and throw it into the gallbladder system, commonly bile duct, up to the fecal matter to make fecal matter flour. It's a process, but the sign of the liver cell must be weighted. Now imagine a alcoholic person who has damaged the liver, and the liver is now cirrhotic, can function at cellular level, and in Zambia, the commonest cancer in Zambian male was liver cancer, hepatoma. Three things were damaging this cell. hapo, especially kachasu, hepatitis B, and aflatoxin, overkept maize, and the ground. You find the liver cells get damaged. So I'm trying to you to relate the cell function can be affected in most of this organ. The smoker, chronic smoke, what do you think about the alveolar cell? The alveolar cells are there to allow oxygen in and allow carbon dioxide out. We call it a alveolar epithelial cell type 1. But to keep this alveoli open, you need surfactant to be produced. So newly born babies, premature ones, they don't have enough alveolar epithelial cell type 2, which is supposed to produce surfactant. So surfactant, again, when it works, it has to be phagocytosed, macrophages. You will be reading this. I want you to relate what we are reading in now. Don't throw away your notes. You need this information. Mistakes we have made in the past. You think you have passed it, you forget about it. I want you to develop a learner. You keep on accumulating knowledge. So as you go higher, you still go back and repair your notes. These notes are very important. So please, my plea that what you are learning now, you need this knowledge for your lifetime. And about the cell health, this is we make all bodies with cells. Cells in the brain, liver, kidney, lung, you name it, the heart itself. So please, I'm saying congratulations. You have done it very well. You are well coordinated. You speak with confidence. Build that confidence. And I want us to clap for the dean, Dr. Chirwa, for this. Well done, Dr. Chirwa. This is the spirit.
And Dr. Chakoma.
Oh, Dr. Chakwa, can we clap for him in a If you have been here participating.
Yeah.
And also all your department, mathematician, chemistry lecturer, and the physics, they should all be participating. Well, this is your faculty job. Because they should also prepare similar presentation. To me, you are already becoming good health workers. Always, when you qualify, you are looking after the sick, you should apply this knowledge. And when you're on your own, talk about it. The more you talk about it, the more you will remember. So the systems, major systems are 7: central nervous system, cardiovascular system, respiratory system, gastrointestinal, number 4, genitourinary, number 5, endocrine system. I think you mentioned some. And then musculoskeletal. Very important, all the 7 systems. Try to relate your knowledge to these major 7 systems. And you come down to the cellular level. That's when we say basic sciences. Even as you are going to be doing a master's degree, you will be using this basic science. Don't lose touch with it. Keep it. You will become good clinicians, good researchers, and good academicians. All these are all yours. Health sector has a lot of opportunity, but we need people who are focused, who are knowledgeable, and starting getting good grades. Avoid to my B pluses, no. You should aim at A plus, B plus, Dr. Chiro. Encourage them. Because at the end of your graduation, you are applying for a job overseas. All they want from you, send us your transcripts. And then you will be comparing from other countries. Transcript, you start making them now. Good grades. So please work hard. Don't play around. This is your time to start building your career. Dr.
Do you remember what I was telling you today about transcripts? Yeah, you are not only competing with your classmates, there are those who went before you. Then we've got people from other universities in Zambia, Southern Africa, and across the world. So employers now are looking at the transcript. Avoid carbon throughout. For A plus, B plus.
That would be very good. Because you apply, people will get from Kenya, Uganda, Zambia, Zimbabwe, they will get transcript. Then the employer is ready their transcript. Which student, which one can we employ? This one has a C, this one has a A. Whom do you think they will employ? So we should start building our career from day one. Work hard, play less. This is what given opportunity. Use your learning time. Concentrate on learning. Not parties every weekend. Please. Avoid that time worth. Concentrate. This is the stage of education. Stage where you dedicate yourself. Believe me, my hardest point of training was 1971 and 72 when I was doing natural science at home. We were doing 2 years, like pre-med. It's a very difficult time because it's about it. You have to read, you have to do practicals, you have laboratories, so a day is short. Don't play with any hour, don't waste any hour. That's the period I slept 4 hours a night to try and catch up. And we did have opportunity of medical school. We only had one medical. So 1972, we wrote the exam, and 600 people, students wanted to go to medical school. They only got 30. Competition was very stiff. Now we have a chance. We have opened. This is our first private medical school. We have opportunities, but use them. Health workers are needed everywhere. What about climate change? Do you think in cellular we have to protect cellular with climate change? Have you seen how drastic it is becoming? The environment is getting polluted and the temperatures are going up. So you as doctor, future nurses, you should prepare to achieve adaptability of human health. Is that okay? Yes. So congratulations. I think we should clap for yourself now. Thank you.
Okay, so before we go, we are going to have a quick online quiz for our presenters and the audience. We'll see if I know we are following.
Dr. Chua, are the questions going to be displayed?
Yeah, it's being displayed.
Wonderful.
Okay, question 1 from the presentation. Which of the following processes requires energy in the form of ATP? Diffusion, osmosis, facilitated diffusion, active transport. Shall we quickly type our answers? From the presenters, D, active transport, we all agree.
Yes.
The frequent attendance, we agree that the correct answer is D. Okay, question 2. You agree? Correct. Wonderful. Question 2, in facilitated diffusion, molecules move across the membrane with the help of A carrier protein or channel protein, ATP hydrolysis, phagocytosis, vesicle formation. Huh? A. Our audience, what do you say? Okay, I can see an A there, correct. We go to question 3. Which statement about osmosis is correct? It is the movement of solid molecules down a concentration gradient. It is the passive movement of water molecules across a selectively permeable membrane. It requires carrier protein and energy. It occurs only in the plant cell. Our audience, what is the correct answer? Someone has written B. Do you agree with him?
Yes, sir.
Okay, B is the correct answer, correct. Endocytosis differs from exocytosis because number one, it requires export materials, while exocytosis import material. Requires energy, exocytosis does not. Endocytosis, it imports materials while exocytosis exports them. It requires a carrier protein, while the other one does not. Which is which? Our audience, we compare audience and presenters. Okay, it involves importation of materials, exocytosis, exportation, correct. Question 5. Which of the following is an example of active transport in a human cell? Active transport. A, glucose uptake in the small intestine using sodium glucose transporter. B, diffusion of carbon dioxide across the alveoli. Movement of water into the red blood cell. Movement of oxygen across mitochondrial membrane. Huh? The audience, we have A, correct. Okay, that marks the end of our quiz. For the students writing 1-1, we have one question which may appear in your quiz. I'm not telling you which one. If we manage to get the questions correct, well and good. So thank you very much. Next week, we are going to have a presentation in chemistry. So to the audience and other students, please go and see the HOD. You'll be given the topic, then we are going to prepare the topic tomorrow. So thank you all for logging in today. Ms. Chinondo, over to you.
Excellent. Oh, this is a great innovation. Dean Chirwa. Interaction, learning, everything. Dr. Chirwa, any comments on any feedback on how you saw how this quiz went?
Yes. Early names better early on, but they quickly settled down.
Pardon?
Next time when they come, they are going to be beyond perfect. Today they are perfect. Next time they'll be beyond perfect. That was a great presentation. They knew the topic, they all understood what they were talking about. And I'm impressed that at this stage in first year, they are able to explain how a disease can come about. They are able to relate what they are learning and the pathologies out there when they are still in first year. By the time they go to preclinical and clinical, they will be superb.
Well done, students. Well done. Well, if we don't have any other questions or comments, we have now. I would now like to invite closing remarks, seeing as how we've come to the end of today's session. I would like to invite closing remarks. I would like to start with our presenters team. Please, presenters, the floor is yours.
From the presenters, we just wanted to say thank you so much to the team for medical sciences for giving us an opportunity. And thank you so much, Professor, for attending and giving us the encouraging words. It really means a lot to us, and we should not throw away the notes, and we'll go back and recite them whenever we need them. So thank you so much, Dr. Chirwa, and thank you so much to everyone who presented. Thank you.
And the participants.
And participants as well.
Excellent. Thank you. Job well done. Well done. I would like to invite our moderator, Dr. Chirwa, your closing remarks.
Okay, once again, I would like to thank the presenters for the great job that they've done. IT and the department really appreciate. They are not been working non-stop on Fridays, including the weekends, but you still had time for us. Professor Lambert, as usual, we appreciate. Could have been anywhere, but you preserve this time to be with the faculty. So we don't take your presence for granted. We really appreciate. For those who do not know, Professor Lambert is our Deputy Vice Chancellor at Lusaka Africa Extension Base for Academics. And is also the founder member and owner of the university. So this university is privately owned by indigenous Zambians. He is one of them. Sure.
Thank you, Dr. Chirwa. I would now like to invite from our professorial team, Professor Lambert, to give his closing remarks.
So on behalf of the Vice Chancellor, maybe the attending, the CEO has traveled out, usually and Professor Peter Mawa is our CEO, Professor Ngeretan is our Vice Chancellor. I say thank you very much. Now start also watching other presentation. Monday is for you, pre-med, and I'm very grateful to the dean and all the department. Tuesday is for preclinical. When you finish pre-med, you are coming to preclinical anatomy, physiology, biochemistry. You should also be watching how they present. Because when you become preclinical, you will be presenting. Then on Wednesdays, we have research. And try to participate and check on the research. Because from this knowledge you are having, this is what is required for research. We have a big research center in Rufunza. You should be developing that interest. Then on a Thursday, we have medical faculty, MVCHB, they present. And it's very, very good. These are clinical cases. You'll be seeing diabetes, they want to be repairing. And Friday, they share nursing, physiotherapy, pharmacy, they share Friday. So every day of the week, at lunchtime, make yourself available. Just to see how we are going to be presenting as you go along. The research aspect, we want you to start developing that in mind. So on behalf of management and the founder members of board, I say thank you very much. Congratulations. This institution is yours. Build it and start praying about it so that it keeps on advancing. Thank you very much.
Excellent. Thank you, Professor Lambert. And I'm sure to all those attending, all of our students have definitely taken those words to heart. We have now come to the end of today's virtual clinical educational grand round. Our Faculty of Pre-Medical Sciences presenters, well done. So please bear in mind, as Professor Lambert said, every day of the week has its own theme. So please stay tuned for more. And for those of you who would like to watch all of our recordings of all of our grand rounds, you'll find them on our Lusaka Apex Medical University YouTube page. So please, as soon as all of our presentations are done, they are immediately uploaded onto our page. So please make your way there. Otherwise, we have come to the end. Please enjoy the rest of your day. Thank you.
Thank you.